At present, the burden of malignant tumors in China is increasing day by day. According to WHO statistics, in 2020, there were 4.57 million new cases of cancer and 3 million deaths in China, accounting for 23.7% and 30% of the global total, respectively, ranking first in the world. The sub-issue of the Lancet published by the research team of the Chinese Center for Disease Control and Prevention pointed out that 65% of cancer deaths in China are male. Top 5 men with cancer: lung cancer, liver cancer, gastric cancer, esophageal cancer, colorectal cancer. Top 5 women with cancer: lung cancer, liver cancer, stomach cancer, colorectal cancer, breast cancer. The traditional way of treating cancer is chemotherapy, but chemotherapy indiscriminately attacks normal cells and cancer cells, and can also cause side effects such as hair loss, nausea, and vomiting. Therefore, people are looking forward to a more precise and less toxic cancer treatment method. At the beginning of the 20th century, German Nobel laureate Paul Ehrlich first proposed the idea of a magic bullet. If there was a drug that could directly locate cancer cells and kill them without harming healthy cells. Based on this concept, antibody-coupled drugs, abbreviated as ADCs, emerged. ADC entered the treatment of solid tumors in 2013, and the US FDA approved HER2-ADC inmetriazumab, T1 German mark, for the treatment of advanced breast cancer. Let's learn about ADC together. ADC consists of 3 parts: monoclonal antibodies, small molecule drugs, and linkers that connect antibodies with small molecule drugs. How does ADC function? 1. The antibody portion of ADC specifically binds to the antigen on the surface of cancer cells, causing the ADC antigen complex to be internalized into the cells, achieving precise localization. 2. ADC is degraded by lysosomes, releasing chemotherapy drugs. Chemotherapy drugs have cytotoxicity and are the components that truly kill cancer cells. Hence, they are called bullets. There are 2 main types of chemotherapy drugs: DNA-damaging drugs and microtubule inhibitors. 3. Chemotherapy drugs bind to DNA or microtubules in cancer cells, inducing apoptosis of cancer cells. When the released chemotherapy drugs are permeable or transmembrane, they can kill adjacent tumor cells, known as bystander effect, further disrupting the internal tissue environment of the tumor and amplifying the killing effect. In addition, the anti-cancer activity of ADC is also involved in the functions of ADCC, ADCP, and CDC. Therefore, ADC can specifically recognize and kill cancer cells, minimizing damage to normal cells and reducing chemotherapy-related side effects. At present, ADC drugs have shown excellent clinical effects in breast cancer, lung cancer, gastric cancer, and other fields. With the continuous advancement of scientific research and technology, we believe that in the future, they may provide patients with more precise and personalized treatment options, thereby achieving better treatment outcomes and improving their quality of life. This is the end of this video. Follow Cheng Yao Biology's video account to bring you more interesting and informative life science knowledge. We look forward to seeing you next time.